All right, this is take number 2 on the first lecture. I learned something important that I can't do the first time, but now we're back on track here, I believe. Before I begin, I want to make sure that you know that you should go to the modules. Go for this one, it's the week 1 module. And if you'll scroll down, you can see the fourth one down here is week 1 lecture material. Now, if you click on that tab, it opens and has the documents for the lecture for this week. So you can see biomolecules and cells, theories of aging, common processes of aging. Those are the 3 topics on which I will lecture this week. And these Word documents are organized as an outline, and I will merely go through and kind of explain it. Sometimes you can go through and understand these on your own, but I always have the recordings here if you would like to follow along with those as well. And if you click on those also, you can download them, you can take notes in them, either on your computer or print them out and write on it, however you would like to do that. And so here's this week, Biomolecules and Cells, which is really a very basic review of biomolecules and cells. Biology majors may or may not need the review, but other students sometimes take this class. It's not strictly a biology class. And so I want to make sure that everyone is kind of on board. We're going to talk about 4 different classes of biomolecules: carbohydrates, lipids, nucleic acids, and proteins. The first is carbohydrates. And there's 3 different types that we'll mention: monosaccharides, disaccharides, and polysaccharides. And of course, you should be thinking, okay, saccharides are carbohydrates. Yes, they are. Monosaccharides are just a single carbohydrate molecule. And even though there are many, many different types of these found inside the body, they're usually made from another molecule. And the ones that we ingest in the diet are glucose, which actually becomes our primary energy source for almost all cells. Fructose, which is more abundantly found in fruits, as well as some other food sources as well. Galactose. Note that we actually convert most fructose to galactose and fructose and galactose to glucose. The liver does that, although some fructose is actually taken into cells and metabolized for different things. Disaccharides, these are just monosaccharides put together. So now you have 2 of them. And the common ones that we consume would be sucrose, and it's really a glucose and a fructose, right? Now that's just common table sugar. Maltose is commonly found in grains. Lactose, you should recognize as milk sugar. Polysaccharides are chains of glucose, and animals and plants store these chains of glucose in different ways. The chains are put together differently. They have a different structure. But that's how living things can actually store carbohydrates for use later as energy, or even as part of a structure, as we'll see in a moment. So animals, including humans, store carbohydrates as glycogen. And this is stored, a lot of it in the liver, a lot in your skeletal muscle, some in cardiac muscle, but only liver can break down the glycogen, and also kidney to a small degree, and break it down into the individual glucose molecules and then release them back into the blood. And that's the purpose, of course, the liver storing glycogen. The muscle storing glycogen is actually, when it breaks down, it's for use within the muscle only. It actually cannot get back out. Plants store glycogen as starch for energy, but plants also form cellulose from glucose, and it's put together in a way that it forms a cell wall around the outside of the plant cell. And when we eat it, we cannot digest that cellulose. We can't digest that cell wall. It becomes part of our dietary fiber. So it goes all the way through our small intestine. Our large intestine will have bacteria that can then break it down, but we ourselves, unless you're counting the bacteria as part of you, cannot break down the cellulose. Lipids, of course, are not water soluble, in general referred to as fats and oils, at least for consumption purposes. Triglycerides is what we typically think of as fat. It's how we store fat. It's a rich energy source. So glucose is our primary energy source, but triglycerides are our primary energy store. We can store far more energy as triglycerides than we can as glycogen. Phospholipids are a lipid with some type of phosphate group attached. They're commonly found in and form cell membranes. They do other things in the body. These are just the primary functions. Sterols are a group of lipids that are built from cholesterol. Okay? They're built on what we call a cholesterol backbone. So they all have a similar structure, and it's what's around the edges that then kind of differs. And that includes the steroid hormones or steroids. They're all made from cholesterol. Nucleic acids are formed from individual nucleotides such as ATP, CTP, GTP, CTP. If you remember AGCT are the four DNA nucleotides, and then U in RNA. But ATP is not just a component of DNA. Of course, it's also the primary energy molecule of the cell. When we said that glucose is the primary energy source, we use the energy from glucose then to actually build ATP and make it as a high energy molecule. DNA, just as a reminder, is double-stranded, found in the nucleus, although we also have some of the mitochondria, just a small amount, 37 genes worth is all. And importantly, it contains the genetic information, which is only 2% of the DNA, which means there's a lot more to DNA than just genes. That's just the important part we often teach, because that's the part we actually understand. We don't understand a lot about the rest of it. And of course, these genes each have codes for making the proteins. The sequence of the nucleic acids will then determine the sequence of the amino acids that are made in the subsequent protein, including information for duplicating cells. The proteins will help to drive that. So now when cells split and divide, they each get a duplicate copy of the DNA that was in the original cell. And of course, we know these are located on 23 pairs of chromosomes, and you got 2 copies from dad, 1 copy from mom. And so you got 2 copies of all of the genes in your DNA. RNA is single-stranded, and of course it's made by reading the code from DNA, and because it's the same language, nucleic acids, this is transcription. And then we read the code from the RNA to eventually make the protein. And those proteins are made from, in humans, 20 different amino acids, and it's the sequence of those amino acids that is critical to the protein function. And letter B here, proteins carry out the vast majority of the body's functions, so those amino acid sequence that was dictated by the DNA sequence is vitally important and can't really be changed too much. Some proteins, their functions can be turned on off. That's probably an overstatement. There's a few where it can be completely on or completely off, but most proteins, it's really more like you're turning it up, the function is turned up, or it's turned down, right? So it's more like a volume control than an on-off switch. And these are just some of the common functions of proteins, getting chemicals in and out of cells because of the phospholipid bilayer, conveying messages inside the cell as well as outside the cell, such as hormones. Many hormones are protein-based. Proteins are also form enzymes that help to catalyze chemical reactions. So metabolism, all of the body's chemical reactions, almost all of them are driven by enzymes, and those enzymes are primarily proteins. Well, now we switch to the cell. Cells, remember, are surrounded by this phospholipid bilayer that is selectively permeable. It's important because then now we can regulate what comes in and what goes out of that cell. Well, oxygen and carbon dioxide, they're actually freely permeable. So that plasma membrane is really not any barrier to them, partly because they're small, but partly because they they readily dissolve in both lipids and water. So the membrane, they just don't care and they zip right through But if you think about it, that's important. So now it's not a barrier to transport of gases, so we can get rid of wastes and have the oxygen come in that we have to have. They're a little permeable to water, not a lot. Water is small enough that even though it's water and is going through a lipid, it's small enough that it can kind of zip through and get through some of them. But every other molecule has to have a protein carrier or channel that'll actually grab something from one side of the cell and put it in or out of the cell, transfer it across to the other side of the membrane. And examples of molecules that need transported would be glucose or sodium. Almost all cells have a nucleus, of course, where the DNA is found. Some cells, such as red blood cells, do not have a nucleus, and therefore they cannot multiply and divide. They cannot make more protein. They can't do repair. So red blood cells, as they get beat up and worn out, which they do because they're squeezing through little tiny capillaries all the time, we're constantly making new red blood cells to replenish them. Point C here, there's so many different kinds of cells in the human body. There are cardiac muscle cells. There are intestinal, what are called the enterocytes that line the inside of the intestines. There are neurons that form part of our brain and nervous system. All of these cells have different functions. They all have different shapes. They have a different look. But importantly, because they all came originally from the same cell, they all contain the same DNA. It's just that some of the genes are turned on in that cell and some genes are turned off, so that each cell will have a different shape and function. But of course, some genes are going to be active in all cells because there's just some genes that all cells need. And that's the end of the first lecture.